It's always an honour to be invited to speak at these meetings, and it's an enjoyable experience, also quite a challenging one, as well as a rewarding one. So hopefully what I'll have to say today will interest everybody, hit the right pitch. I'm aware that there's a mixture of people in the room, some of whom know more about the disease than others, and I've tried to pitch it somewhere in the middle, so apologies in advance to those of you who know lots about it, for whom it might be a bit simplistic, and vice versa to those of you for whom this is a disease that you don't know much about. I hope it's not too complicated. Anyway, we'll see how it goes. So I've got this title really that I quite like. There's obviously when we doctors are very good at complicating things, but actually life's actually not that complicated if you distill it down to its component parts. And I think there's a way of explaining this in simple terms that you'll hopefully identify with. So I have to do the boring bit first, I'm afraid, just to sort of set the scene. And again, I guess lots of you know all this already, in which case I apologise. So CLL is the commonest adult leukemia in terms of the number of people alive who have the disease. The actual rate at which it occurs is there's 5 new cases per 100,000 people per year, and that means there's about 3,000 new cases per year in the UK, but many more people living with it, obviously. It affects males more frequently than females, and interestingly, it's less common the further east you go in the globe. And if you go to the Far East, it's not that common at all. This is a genetic thing, and we don't fully understand it. It's predominantly a disease of older people, but by no means exclusively. And the youngest patient I ever came across was in her 20s when she had it. So, but it does predominantly affect older people. And of course, as you get older, you pick up other medical problems, you become more frail, and that poses challenges for the treatments sometimes as well. So that's a relevant fact. So this is what the disease looks like down the microscope, and for those of you who've never looked down a microscope, I'll point out the following cells. So these are the red blood cells that transport oxygen around the body. Those little ones there are the platelets. The platelets, they're the little sticky ones that stop you from bleeding when you cut yourself. And these are the white blood cells. And the issue here is that this blood film has too many of these white blood cells, and they're all the same. And this is a fairly typical picture of CLL. The cells, they're pretty boring looking to an experienced haematologist. They're little small things with most of which it consists of the so-called nucleus. And some of them get squashed when the slide's prepared. They're mechanically fragile, so when you smear them on a glass slide, some of them get squashed like that one there. So this is a very typical appearance. So you can suspect the diagnosis on a blood film, but to prove the diagnosis, you need to do some fancy tests. And the basic test that we do to diagnose CLL is it's called flow cytometry. And I'll talk you through it because it's quite interesting really and quite clever. So you essentially take the patient's blood and you react the blood, you mix it with a mixture of antibodies. And antibodies are proteins that stick onto things. And you can label antibodies with different molecules. And in this technique, the antibodies are labelled with chemicals called fluorochromes. And fluorochromes have the property of absorbing light energy and re-emitting it at a slightly longer wavelength. And you can exploit this by detecting the presence of the antibody on the surface of a cell that it's stuck to if you expose that cell to laser light. So you take the blood, mix it with these fluorochrome labelled antibodies, and then put it in this machine. There's a little nozzle there, so the tube of blood goes into that machine, and the machine processes the blood, very, very, very clever fluidics, and it produces a single, it basically squirts the cells into a single file. All these cells come down this very, very fine nozzle in a single file and cross this laser beam. Okay, it's an ultraviolet laser beam, and the laser beam excites the fluorochromes that are stuck onto the antibodies that are stuck onto the cells, and each fluorochrome emits a different coloured light, which the machine then detects. So, and you can do this with a mixture of antibodies, each of which is linked to a different light. And this is the sort of readout you get from the machine on the computer. And here, for example, you've got an antibody that sticks to this molecule called CD19. That's a marker of B cells, so-called B lymphocytes, and CLLs are B lymphocytes. And you get this signal here, on this axis here. CLL cells also emit, they also express this molecule called CD5, which is normally expressed on T cells, but they aberrantly express it. This is one of the features of CLL cells, and you can detect that. And here, FITC is the green colour fluorochrome, and P is the red one. So this is green light, and that's red light, because the antibodies are stuck onto fluorochromes that emit those colours. So by including a mixture of different antibodies and building up this picture, you can understand what's on the surface of the cell and diagnose the disease accordingly. And CLL has this very typical profile of cell surface markers that you can detect in this way. So that technique is called flow cytometry, and it's the heart of CLL diagnosis. So we all know that the abnormal cells accumulate in the bloodstream, but they really grow in the tissues, and one of the tissues that they particularly like to be in is the bone marrow, and that's obviously why we do bone marrow tests. And I guess many of you have probably had bone marrow. I hope it wasn't too uncomfortable an experience, but basically you lay the patient on the side and pop a hollow needle into the back of the hip bone, and you can extract the bone marrow both in liquid form. So you apply suction, and some of the cells that live in this middle space in the bone marrow are pulled out into the syringe, and then you can smear them on a slide just like you would a blood sample. Or you can take a solid piece of bone marrow by using a slightly different needle with a sort of cutting blade at the end, wiggling it around a bit, and then when you pull it out, it pulls out a piece of bone about the size of half a matchstick, and then you fix that in formalin and embed it in paraffin, and then slice it up, and then you can look at it. It's like sort of sliced boiled eggs, whereas a blood film is more like looking at fried eggs from the top kind of thing. So that's how the bone marrow is done. It sounds horrendous, but I guess those of you that have been through it, it's not pleasant, but it's not as horrendous as I probably made it sound. This is what the bone marrow looks like. This is when you take a solid piece and slice it up and stain it, and what you can see here is several things really. You've got these fat spaces, and this is a feature of healthy bone marrow. So a lot of it is fat space, and then in the bit in the middle here, which contains the cells, there's a whole mixture of cells going on there. You can see these big ones; those are the so-called megakaryocyte cells that make platelets. The platelets bud off of these cells, and then those ones with the very dark nuclei, they're the immature red blood cells that eventually lose their nuclei and then release the cytoplasm as red blood cells. And then in there you've got these sort of you can't really see it at this power, but when you look closely there, they've got granules in. Those are the granulocytes or healthy white blood cells. And there's a whole mixture of things, and there's fat. So that's a healthy looking bone marrow. This is what you get in CLL. So first of all, there's many more cells here. So the cellular component of the bone marrow kind of expands at the expense of the fat spaces. So there's too many cells there. That's the first thing. And when you look at these cells, they're all the same. You don't get this kind of mixture of different cells that you see in healthy bone marrow. So, and what's happening here is that the CLL cells are just multiplying in the bone marrow and overrunning it, basically. And this is a problem because the function of the healthy bone marrow is to make healthy blood cells. So if there's CLL in there overrunning the bone marrow, the bone marrow as the factory, if you like, that makes the healthy blood cells doesn't work properly, and you run out of especially red blood cells and platelets. Okay, and that's one of the reasons that CLL causes a reduction in red blood cells and platelets especially. For reasons we don't fully understand, the healthy white cells seem to be relatively spared. They can fall in CLL, but they don't fall as much as the red cells and platelets for some reason. Okay, so the CLL cells, they build up and grow in the bone marrow, and they also build up and grow in some of the body organs as well. And the liver and spleen are two of the sites that these cells like to be in. So this just shows there's the liver there and the spleen. And the spleen can get really, it's supposed to just fit under the ribs there. In a healthy spleen is very difficult to feel. You can't feel a healthy spleen, in fact. But when it grows, it sort of first of all peeps under the left ribs and then can expand right down to the bottom of the abdomen and become very large. And if you're wondering what that picture there is, it's a sponge. And one of the things that happens when the spleen becomes enlarged, it acts as a kind of sponge. It soaks up all the healthy red blood cells and platelets and lowers their levels in the blood. So having a big spleen is another reason why patients with CLL can become anemic and have a low platelet count. So the other place that CLLs accumulate is the lymph glands, and that picture there shows this man here with enlarged lymph glands in the neck and under the arms. That's quite an extreme case. And if you remove a lymph node and slice it and look at it under the microscope, this is a healthy lymph node, and a bit like the bone marrow, there's lots of structure there. There's a mixture of cells. There are these things called secondary lymphoid follicles with this pale area in the middle. This is the so-called germinal center. This is a lymph gland which is doing its job. It's responding to things in the body that shouldn't be there and reacting against them. That's going on all the time and keeps us healthy. And this is what happens in CLL. The lymph gland fills up with these cells. So if you didn't know that was a lymph gland, it would look very similar to the bone marrow picture that I showed earlier. The CLL cells are just overrunning it, and therefore stopping the lymph glands from doing their job properly, which is to fight infections. So one of the problems that you get in CLL is this susceptibility to infection because it overruns the immune system, the healthy immune system. You can also get what I've called abnormal immune reactions as well, where the CLL cells kind of subvert the immune system and cause it to do things it's not supposed to do, like fight red blood cells and platelets, and we call that autoimmune hemolytic anemia, or ITP, immune thrombocytopenic purpura. All that means is that the immune system's attacking the red blood cells and platelets and lowering them, and that's another reason why patients with CLL can become anemic and have low platelets. Okay, so this is something you could probably teach me about, symptoms of CLL. So in my experience, the really predominant symptom that patients with CLL experience is tiredness. That's number one, head and shoulders above anything else. And of course, it sort of creeps up on you. You don't realise how tired you are from CLL sometimes until you treat the disease, make it better, and eventually, once you've got over the side effects of the treatment, your energy levels come back. It's all a bit non-specific, of course, and other things can make you tired. But that, from my perspective, anyway, and you know, I invite you to agree or disagree. Tiredness is one of the main features of this disease. As the disease grows, you can get other features. You normally only get these things if there's lots of disease on board. You can get sweating, you can get fevers, and you can get weight loss as well, because the growth of the CLL cells consumes all of your energy, and the cells release chemicals that suppress your appetite and make you lose weight. So those are the classic features of CLL itself. But you can also get complications, as I've alluded to. So here, this picture here, one of the ways to detect anemia is to look in the eyes, and if the conjunctivae look very pale, that's an indication of anemia. And there's a hint of yellowness here as well, which may suggest that this patient here has autoimmune hemolytic anemia. The immune system's destroying the red blood cells. And when that happens, all the hemoglobin, the pigment, if it overloads the liver's ability to deal with it, it builds up in the blood as this yellow compound called bilirubin that makes your eyes go yellow and your skin. So that's anemia is a very common finding in CLL, and this is what happens when the platelets go down. You get these little, they're tiny little bleeds under the skin from the little blood vessels, the capillaries under the skin, and we call it purpura or petechiae. And this is quite common, but you have to have really low platelets for this to happen. You don't really see it until the platelets go below about 20 or 10 or so. This here is shingles, and shingles is quite common anyway, but it's even more common in CLL, and it can be quite nasty. I guess many of you may have had it, and it can cause nasty acute rashes and leave this dreadful lingering pain behind. It's like electric shock sort of pain for a long time. So it's quite a nasty condition, and of course it reflects the fact that the immune system isn't working properly. And what it actually is, it's the chickenpox virus. So virtually all of us have chickenpox. Usually we get it as children, and most of us don't realise we've had it even. And when we have the virus, it stays in our bodies for life. And in health, the immune system controls it and stops the virus from causing any subsequent problems. But if your immune system is suppressed, the virus can wake up and cause problems. And it hides away in the nerve roots, in the spinal cord. And when it wakes up, it travels down one of the nerves onto the skin and then appears on the skin in a very characteristic patch that corresponds to the area of skin that particular nerve feeds. So that's why you get shingles in a particular place. And in this case, it's around the eye. That's called ophthalmic shingles, but you can get it anywhere in your body. But it's usually a very discrete patch for the reasons I've outlined. And this slide here shows a pneumonia. So this is what a chest X-ray looks like. These dark areas are the lungs, and you can see the middle of the right lung has this. It looks white rather than dark, and this is an area of what we call consolidation, and that's typical of a pneumonia. And again, that's a reflection of a non-functioning or poorly functioning immune system. Okay, so that's the kind of whistle-stop tour through CLL, the basics of CLL. And now I'd like to come on to the issue of variability. So what's really striking about CLL is that although it looks pretty much the same down the microscope, those cells I showed you are pretty much the same in all patients. It behaves very differently in terms of its clinical course between different patients. So there are certain things that can predict the kind of big picture if you've got newly diagnosed CLL. So one thing is how much disease there is at diagnosis, how much it's already grown by the time it presents. Okay, and we call that clinical staging, and I'll go into that in a little bit more detail on the next slide. And the other determinant of outcome that's based on very simple things is how quickly it keeps growing after you've diagnosed it. Okay, so there's how much it's already grown when you diagnose it, and then how fast it keeps growing afterwards. And we can measure that by looking at the blood lymphocyte count and measure how quickly it goes up. And there's a measurement called the lymphocyte doubling time, which is simply the time taken for the lymphocyte count to go from 10 to 20 or 20 to 40 or 40 to 80. And you need to do more than 2 measurements to get the picture because it can fluctuate. But if you do 3 or 4 measurements over time, you can draw a graph basically and work out the doubling time, and that tells you how fast the disease is growing. Okay, so this is clinical staging, which is just a reflection of how much disease there is on board. And there are 2 systems that are applied. There's one European system called the BNA system, and another one that's used mostly in America, which is called the RIE system, but they're essentially the same. So we tend to use the BNA system on this side of the pond. And just to summarise, stage A means essentially that all you can see is the disease mostly just in the blood. Okay, so it's not causing that much in the way of swollen lymph glands or liver or spleen, and it's not suppressing the function of the bone marrow too much. So that's what we call stage A disease. Stage B is where, yes, it's in the blood, but it's also causing some swelling of either the lymph nodes or the liver and spleen, and you have to have at least 3 sites of involvement, and the sites are the neck, under the arms, groins, liver, spleen. So you've got 5 to choose from. You can do all this without doing any scans. You can just examine patients to work this out. But importantly, although it's in the lymph glands, the bone marrow function is maintained. So the red cells are okay, the platelets are okay. Stage C is basically when the bone marrow starts to fail. There's so much disease affecting the red blood cells and it's affecting the platelets, irrespective of what else is going on. That's called stage C. And this is a very old paper, but this just shows you, at the time the study was done, of course treatment's come on since then, but at the time that the study was done, these are the average survival times corresponding to these different groups of patients, separated by how much disease there was at the time of diagnosis, how much it had grown at the point of diagnosis. Okay, this is lymphocyte doubling time. So this is how much the disease carries on growing after you've diagnosed it. And this is a plot of lymphocyte count with time, and it usually goes up exponentially, and then you can work out from the graph the time it takes for the count to double. And then when you separate patients into whether it's what we call smouldering CLL, whether it's a slow lymphocyte doubling time or a faster growing version, that affects survival. So the question arises, why do we need these fancy tests? So we can already make predictions about survival based on the things I've just mentioned. The trouble is, if you just take clinical staging, tumour burden at diagnosis, how much it's already grown, yes, it can predict survival, but it doesn't predict very much else. Furthermore, the same can be said of lymphocyte doubling time. And the trouble with lymphocyte doubling time is that to measure lymphocyte doubling time, you have to sort of wait. You have to wait and measure it over time. And sometimes that takes a couple of years. So if you're a patient with newly diagnosed CLL and you want to know what the future holds, and especially if you've got early stage disease without too much going on at the time of diagnosis, you have to wait to find out, and that can be improved on. Furthermore, so that's an issue for patients largely, but doctors also want help with treatment decisions as well, and things that just predict how long you might live for are not really helpful when it comes to treatment decisions if you've got a choice of different treatments and things to do. So we need tests that give us more specific information that can help us with specific treatment decisions. That's the answer to the question. Okay, so if we just take the patient's perspective for a moment, and especially patients who are diagnosed with low tumour burden disease or early stage disease and want to know something about the big picture, what tests can help? So there are basically two tests looking at the chromosomal abnormalities, and I'll elaborate on that in a bit. And the other is understanding the cell of origin of the disease, and again I'll elaborate on that as well. And there are tests that we can do, and you may have heard of this test called FISH, which stands for Fluorescence, in-situ hybridisation, and I'll cover that in a second. And these four abnormalities that are the most commonly observed chromosomal abnormalities, and they can have they can give quite useful information in terms of the big picture. Similarly, with cell of origin, we use this test called immunoglobulin heavy chain gene mutational status. I apologise for sort of burdening you with jargon, and hopefully I'll explain it a little clearer in following slides. But if you combine these two tests, you can essentially stratify patients into two groups: those that are going to do reasonably well, and those that are going to do less well, and somewhere in the middle. And that some patients don't want to know, and that's fine. For other patients, it's really helpful knowing what the future holds because it allows you to plan things. So that's the kind of overview from a patient's perspective. So just focusing on chromosomal abnormalities and FISH. That's the fluorescence in situ hybridisation. What this is all about is looking for chunks of chromosomes or DNA that are either missing or amplified. In other words, there's an extra copy of the chromosome. And to do that, you make a smear of the cells, and then you stain the cells. Instead of staining with an antibody, you stain them with a probe, again, that's fluorescently labelled, that sticks to specific bits of the cell's DNA. And here you can see, so this upper panel here has been stained with two probes. One region of the DNA called 17, the short arm of chromosome 17, and the other binds to the long arm of chromosome 11. And these are two bits of DNA that can be lost in CLL and which have prognostic importance. And this is a healthy cell here so those green dots correspond to the 11q bit of DNA, and the red dots correspond to the 17p bit of DNA. That's a healthy cell. So this one here, you can see there's two green dots but only one red dot. Okay, so this cell here has lost that bit of DNA on chromosome 17p. Similarly, this one here, you got two red dots. There's two copies of chromosome 17, but only one copy of chromosome 11q. And healthy cells are supposed to have two copies of everything. Yeah. So this cell here has lost that chunk of chromosome 11. So this is an 11q deleted case of CLL, and this one here has lost the chunk of chromosome 17p, which is so this is 17p deleted CLL. And you can do a similar thing with other chromosomes. The other two that we look at are the 13q, the long arm of chromosome 13, and trisomy 12. So some cases have an extra copy of trisomy 12, and you can see that here. So trisomy 12 is you can see the chromosome 12 is labelled with the green dot here. So that's 1, 2, 3 copies there, and 2 copies of the chromosome 13q. And this one here, you've got 2 copies of 12, but only 1 of 13q. Do you see? So, so we can categorise patients according to the chromosomal abnormalities that they have. And when you do that and look at survival over time, the groups spread out. So again, this is an old study, and treatments have come on, so wouldn't pay too much attention to the absolute figures here. But those patients here, these are the 13q patients that do particularly well. These are patients who either have none of these things or the trisomy 12, who are kind of average. These are the 11q deleted patients who do a bit worse, and those are the 17p deleted patients who do worse still. Graph. Well, this is the proportion of patients who are alive over time. Yeah. So it's quite complicated, but also quite important. Okay, so remember there were two things that help to give the big picture, the survival type data. One is the chromosomes, and this is now we're moving now into the cell of origin. Okay, so let me just take a sip of water. So where does CLL come from? You know, how does that relate to normal immune cells? So this diagram here, it looks complicated, but actually quite simplistic. But so this shows a normal immune reaction. Okay, so what happens is you have a B cell here that encounters. So all B cells, the job of a normal B cell is to react against the antigen to which it reacts. And there's a very clever process that means that each of your B cells in your body, you've got billions of them, reacts to a different antigen, okay? And the receptor that binds to the antigen is very cleverly engineered by the body to have this incredible diversity. So when the antigen comes along that sticks to a particular B cell, it stimulates that cell to move into, you saw those pictures of, remember the lymph node I showed with those so-called follicles and the pale area in the middle. So this is a kind of a schema of what happens in those follicles. So the B cell finds its antigen from amongst all the billions of antigens that are out there, moves into that clever bit of the lymph node, and then undergoes this reaction, okay, which requires contact with other types of cells. And it comes out the other end as a fully fledged cell that makes an antibody that reacts to the thing that stimulated it in the first place. Okay, and that antibody then gets rid of the thing that stimulated it in the first place. So this is what happens in your normal immune system. It makes antibodies that fight things that shouldn't be there. So, and then there's another pathway that bypasses that reacts to a slightly different set of antigens. Anyway, how does this relate to CLL? Well, for each of these cells here, there's a cancer equivalent. Okay, so cancer arises when a cell goes wrong and starts to multiply itself in an uncontrolled way. So each one of these cells has a cancer equivalent, and we think that the CLL cells are the cancer equivalent of two different types of cells in this process. One coming from a fairly early stage during all this reaction process, and one coming from a much later stage. Okay, so there's two different types of CLL depending on how the CLL cells relate to what's going on in the normal immune system. And we can detect doing this thing called immunoglobulin gene mutation analysis. So you look at the receptor on the surface of the cell that sticks to its antigen, and by analysing it, you can see mutated and unmutated. Okay, I'm sure you've heard this expression, mutated and unmutated. Now, mostly when genes are mutated, it's bad. It sounds bad, doesn't it? But with the immunoglobulin gene, mutated is good. So it's this opposite way round to what you might think. It's kind of counterintuitive. CLL patients over time separated by whether their immunoglobulin gene is mutated or unmutated. Okay. So, just to summarise, we talked about simple things, how quickly how much the disease has grown by the time of diagnosis, how much it carries on growing. We've talked about fancy tests that can give information on the big picture, the overall prognosis. And we've talked about chromosomal abnormalities detected by this technique called FISH. And we've talked about the cell of origin of CLL detected by this technique called immunoglobulin gene mutation analysis, mutated or unmutated. Okay, and that helps patients. If you have this information as a patient, it allows you to. It gives you big picture information and allows you to some extent plan your life. And as I said before, for some patients that's a useful thing. Others prefer not to know. That's fine. Okay, from a medical point of view, from a doctor's point of view, we want a bit more than that. We want tests that can help us make or advise on clinical decision making. And that's what I'm going to go on and talk about now. Okay, I'm glad you like the slide. So there are essentially three, as a doctor, advising a patient with CLL on treatment, there are three main questions to ask. The first is when does treatment need to be started? The second is what treatment to give. And the third is what to do once treatment has finished. And as I hope I can explain, some of these fancy tests can help with that decision maker, with all three decisions. So I'm going to start with the second one actually, because it's actually a more logical place to start. What treatment to give? So this slide, I think it's a concept that many of you will be familiar with. So if we're talking about initial treatment, so we're talking about patients that have never had treatment before and they're facing their first treatment, they need treatment, and there's a choice. And currently the choice involves, it's all essentially the same type of treatment. It's what we call DNA damaging chemotherapy based treatment. In other words, it's drugs that kill the CLL cells by damaging their DNA and eliciting a kind of suicide response from within the cell. And there are different types of chemotherapy that we use. So FCR, for example, I'm sure I know for sure there are people in the room that have had FCR. So the FCR is very effective. It's also quite challenging as a patient. It's quite intensive and has quite a few side effects that need careful management. On the other end of the spectrum, we have chlorambucil. It's been around for 50 years, but it's still a very useful and effective drug. It's not as effective as FCR, but it's much better tolerated. And in the middle, we've got bendamustine. And these days, we usually add what we call a CD20 monoclonal antibody to the chemotherapy. And the prototype for that is rituximab. So the R in FCR is rituximab. So it's an antibody, a drug-type antibody that sticks onto the surface of the cell and tricks the immune system into thinking that it's dealing with an infection, and then the immune system helps to eradicate the thing that the antibody is stuck onto. But there are other antibodies as well. So there's ofatumumab and obinutuzumab, and they all pretty much act in roughly the same way and enhance the action of chemotherapy when you combine chemotherapy and the antibody. But the point I'm making here is that it's all chemotherapy-based treatment. There's a choice. At the left-hand end, you've got the more effective but more intensive and challenging treatment, and on the right-hand side, you've got the slightly less effective but much better tolerated treatment. And the idea is to tailor the treatment according to the patient that you're advising. So. And age is not really a barrier here. So you have some older patients who are really fit, and they can probably tolerate FCR. Equally, on the other side of the spectrum, you have some fairly frail patients with lots of other medical problems, and on balance, you might judge that FCR there's a risk of it doing more harm than good. And for these patients, a gentler approach is more appropriate. There's always a choice, and this always needs discussion. But this is the kind of basic paradigm within which basic treatment decisions around choosing the type of chemotherapy is made. Trouble is, not everybody responds to chemotherapy-based treatments. So this again shows instead of survival, this is what we call progression-free survival. So this is the proportion of patients who are alive and in remission over time. And this is from a trial, the German CLL8 trial, which was the first big trial to show the benefit of adding rituximab to FC chemotherapy, fludarabine cyclophosphamide chemotherapy. And it led to the change in clinical practice, the adoption of FCR as the standard of care for fit patients. And here, patients have been separated into groups by chromosomal abnormalities. You remember those fish tests I took you through with showing up the little dots showing the bits of missing DNA and so on. So what you can see here is they kind of mostly sit together. The lines mostly go together. But this one here sticks out like a sore thumb, okay? And surprise, surprise, this group of patients is defined by having the 17 deletion. So there's a chunk of DNA missing on the short arm of chromosome 17. So although FCR generally is a really good treatment, it's not such a good treatment for this group of patients. So the question is, what's going on chromosome 17p? What is it about this bit of DNA on the chromosome that's so important? So what I've shown here is a normal cell and a CLL cell with a 17p deletion. And these are the two copies of chromosome 17p in the normal cell. And this red bit here is a gene, and genes are it's the genetic code that encodes proteins that are expressed by the cell. P53. When it's a protein, you drop the T and just call it P53. So you've got two copies of the gene, and the cell converts the genetic code into a kind of intermediate form called messenger RNA, okay, and you get that produced from both copies. And then that messenger RNA is then converted into the actual protein that does the function, has the function, okay? Proteins are the kind of building blocks of the cells that actually do stuff, if you like. If you look at if you analyze a cell that has a 17p deletion, there's the normal chromosome there, but there's this chunk missing. Okay. Furthermore, the copy of the gene that's there it's not normal. It's abnormal. It's got what we call a mutation. So it's there, but it's abnormal and it doesn't work properly. So you get the messenger RNA produced, and that's converted into protein. And in fact, the mutant P53 protein is actually more stable than the normal version. So there's more of it, but it doesn't work properly. It doesn't do its job. Okay. It's a little bit more complicated than that because you can get either a deletion or a mutation. Mostly they occur together, but sometimes you can get one or other on their own. And in practice, it has the same effect, biologically and clinically. So to pick up all of these abnormalities, you need to do both tests, basically. This is a test that I haven't mentioned yet. This is looking at bits of DNA that are missing. This is a slightly different test that involves what we call sequencing to read the genetic code. So what is it that P53, wild-type P53 does, the normal protein, that's so important? It's basically the good guy in the cell, okay? It's the superhero of the cell. And what it does, so when you expose when you treat a patient with chemotherapy, the chemotherapy gets inside the cells and it causes this P53 protein to become activated. It's like kind of wakes up. And when it wakes up, it basically regulates the expression of a whole bunch of other genes, okay, 200 or so genes. And these other genes fulfill these three functions. So first of all, they induce the cell to commit suicide. They kill the cell. And they stop if there are any cells that are still alive, they stop them from growing. And they also have this kind of longer term, more subtle effect. They keep the disease stable. They stop it turning nastier with time. All cancers have this kind of predisposition to acquire new changes that give the cells a growth advantage over their neighbours. It's a bit like sort of Darwinian evolution in a single patient. And this gene here keeps everything in check, not just in terms of growth, but also in terms of stability over time. So you can imagine what happens when you lose p53 function is you lose all these effects here. So chemotherapy doesn't kill the cells, it doesn't stop them from growing, and the disease tends to evolve more rapidly and acquire new changes that make it nastier. Okay, you lose the good guy. Fortunately, we do have drugs that can kill CLL cells and perform these other functions here independently of p53. They can bypass the problem. Okay. And that's really useful. It means that we do have treatments that can work in these patients for whom chemotherapy is not such a good treatment. And to fulfill these criteria are, first of all, very simple treatment like steroids. I've seen some amazing responses with just prednisolone, actually, in some patients with p53 deletions. We used to use Campath a lot, and I guess there are some of you, some old-timers, as it were, in the room who may have received Campath. In fact, I know there are. That's an antibody drug, very effective, but quite challenging in its way. There are drugs like lenalidomide that work in a slightly different way, both direct and indirect mechanisms of action. We've got anti-CD20 antibodies like ofatumumab, which can be used on their own. And then we've got this new generation of treatments, which are probably, well, almost certainly better than these other ones, ibrutinib, idelalisib, and venetoclax. So these are the three really exciting new drugs that are now around, which, again, they produce all these effects, but don't require p53, so you can bypass the blockage in the system. Okay, so I've mentioned these drugs. I think no talk about CLL these days will be complete without a little bit of elaboration. So ibrutinib and idelalisib, these are the two prototype drugs which from an entirely new type of entirely new class of drugs that target components of the so-called B-cell receptor signaling pathway. BCR stands for B-cell receptor, and I've mentioned this receptor before. It's the structure on the surface of the CLL cell that you can analyse to determine where the cell is originated from, the cell of origin, and the B-cell receptor plays an absolutely crucial role in the biology of CLL. It transmits signals both from the cell's environment, and also there's also evidence that even without stimulation, the receptor generates intrinsic signals, and those signals basically keep the cells alive, help them to grow, help them to resist attempts to kill them with chemotherapy and so on. And the ibrutinib and idelalisib are drugs that block specific components of this signaling pathway. So all these proteins kind of talk to each other and produce effects. And when you block, so ibrutinib blocks this protein. You can't really see it that well on the slide. It blocks this protein called BTK, or Bruton's tyrosine kinase. And idelalisib blocks this enzyme called PI3 kinase. And both of these proteins fulfill an absolutely crucial role in this signaling pathway. And when you block them, the signaling pathway no longer works, and all the functions that the signaling pathway fulfills no longer apply. And the cells kind of basically curl up and die over a long period of time. That's roughly how it works. And of course, they also, these two drugs also have the effect of redistributing the disease from one body compartment to another. So one of the striking features of patients treated with either ibrutinib or idelalisib is that the cells move into the bloodstream from the bone marrow and the lymph nodes. So you see a very dramatic shrinkage of lymph nodes, and the blood count improves because the bone marrow is being cleared out. But the white blood cell count goes shooting up, and you might, if you just looked at the white blood cell count, you might think the treatment wasn't working. But in fact, what that represents is an anatomical redistribution of the disease from the tissues into the bloodstream. So it's quite interesting. And these CLL cells don't really like being in the blood. They don't get the support that they need in the blood that they get in the tissues. So moving them into the blood is part of the reason, part of the way that these treatments actually work. And I think it's true to say that these two drugs, ibrutinib in particular, have completely revolutionised the treatment of CLL. Definitely in patients that have had previous treatments, but increasingly in previously untreated patients who have a p53 problem. And that's one of the reasons for looking for it in the first place, to identify patients who might benefit from this kind of approach. Okay, venetoclax is the third really exciting drug that's around, and this is different again. Okay, so and I'll just talk you through this because it's very interesting. So this slide here shows that this is a mitochondrion. This is called a mitochondrion, and these are structures or organelles inside of all cells that basically make the cell's energy. Okay, but they also fulfill other functions, and they basically regulate whether the cell. They make a decision, they make life or death decisions for the cell. Okay, and there are these proteins. So one's called BIM and one's called BAX, and they're like the cell's cyanide capsules, if you like. Okay, if they're released, they kill the cell, and they do that by binding to the mitochondria, and then they punch holes in this outer mitochondrial membrane, and then the mitochondrial releases this thing called cytochrome C into the cytoplasm, the cytoplasm of the cell, and that's fatal from a cell's point of view. It causes all sorts of chaos that means the cell dies. Okay, so the cells, every cell in our body is equipped with these cyanide capsules, effectively, but the cyanide capsules are locked in a cupboard. Okay, and that cupboard is called BCL2. Okay. And what venetoclax does, it kind of unlocks the cupboard. Okay, it removes the cyanide capsules from BCL2 and allows them to stick to the mitochondria and induce the release of cytochrome C, which kills the cell. So this is all very clever. The really amazing thing is that this is not, this process happens in all cells. Okay, yet CLL is somehow exquisitely sensitive to venetoclax. And in fact, venetoclax, if you give venetoclax, its side effect profile is remarkably low. It doesn't cause that many side effects, which is amazing considering what it does and the fact that all cells are equipped with this mechanism. There are obviously other mechanisms that stop this from happening in most cells, but CLL cells seem to be exquisitely dependent on these interactions between BCL2 and these two cyanide capsules. So there's some sort of specificity there that means this is a really good drug because it works really well in CLL but doesn't cause any collateral damage. That's more serendipity than anything. But it's a very, very interesting, very effective drug. So what's the current situation with regard to availability of these drugs through the NHS? So these are the current NICE indications. So basically, idelalisib is available. And the thing about idelalisib is that you because of the way that the trials were done, you give it in combination with rituximab. So you don't give it on its own, you give it with rituximab. And the thing I forgot to say about all of these three drugs is that they come as tablets, okay? So they're not IVs, they come as tablets, and you take them and carry on taking them for as long as they carry on working, okay? Just like an aspirin or blood pressure tablet. So in that sense, they're very different to chemotherapy. But idelalisib you do need to give with rituximab. So idelalisib is available for previously treated CLL if it's come back within 2 years of the last treatment. And it's also available for patients who've not been treated before if they have either this 17p deletion, that bit of chromosome missing on, that involves the p53 gene, or if there's a p53 mutation present without the deletion. The same is true of ibrutinib. So ibrutinib is basically available for exactly the same indications, except there's no time limit on the time between previous treatment and relapse. So these are drugs that are already available, including in previously untreated patients if you've got one of these abnormal test results, which means that to access these in the front line, you need to do the tests in the first place to know whether you fulfill these criteria. Venetoclax has got a bit stuck. It was initially rejected by NICE earlier this year. There was a further committee meeting in March. I mean, some of you probably I'm sure there's somebody in this room who probably knows more about this than I do because you may have been involved in that meeting. The final outcome is expected in June. It has a license, so it's you know, legally it's available, and basically, again, you see, it's licensed for patients that have already been through a B-cell receptor antagonist. So you have to have gone through either ibrutinib or idelalisib to get to venetoclax. But essentially, otherwise, the indications are the same and include having a 17p deletion or p53 mutation. So there's a kind of theme emerging here, I think we'll agree, that to access these new drugs early on in the disease course, you need to do these tests to find out whether you have a p53 deletion or mutation. So this brings me to, so I apologise again because I've bombarded you with loads and loads of information. But if we try and distill everything down into a take-home message, if we come to the patient who needs treatment, the question is what treatment? What's the best choice of treatment? How can tests help? So basically, you need to do FISH to look for a 17p deletion, and you need to, it's not on the slide actually, I apologise for that, but you also need to do sequencing to look for a p53 mutation as well. And if you've got either of those things, non-chemotherapy drugs are appropriate and are available. If you don't have either of those things, then a standard chemotherapy-based approach is appropriate. So these tests are of practical value in terms of making decisions about which treatment to have. That's the take-home message. So we've dealt with that question. So moving up a question, when to start treatment? So I guess everyone's familiar with the concept that this disease there's no evidence that treating patients who don't have any symptoms or complications doesn't do any good. Okay, so we reserve treatment until we think it's needed, until there's something to treat. And some patients can go on for years, decades even, perfectly well and not need any treatment. And if you were to treat those patients prematurely, you wouldn't be doing them any favours. Yeah. So that principle still applies, but there may be some patients within the overall group of patients with what we call asymptomatic early stage disease. There may be subsets of patients who might respond. Furthermore, we don't. All the old studies were done with old-fashioned drugs. Now we've got newer drugs. There are open questions about whether earlier treatment with the newer drugs does any good, especially in those patients who are at higher risk of the disease growing quickly. So, and there are obviously trials ongoing to address these questions. And the question arises, what tests are what tests are currently available that may be of some use outside of trials to help with the decision-making process about when to start treatment. So if we knew that a given patient was going to have a very gentle clinical course and not require treatment for a long time, not have any complications or symptoms for a long time, we could adapt that patient's follow-up. We could make it easier for the patient by seeing them less frequently, even doing remote consultations by telephone, just by adapting the follow-up so it's proportionate with the problem in hand. Equally, if we worked out that a given patient was going to have early problems with their disease, we might want to follow them up more closely so we can anticipate those problems before they became more difficult to deal with. And the sort of problems that it would be really nice to predict, first of all, is it's nice to predict how quickly the disease is going to grow, and it's also nice to predict the risk of the disease changing into something nastier like Richter's transformation. If we knew which patients had a higher risk of those two things, we'd be able to keep those patients under much closer observation and be in a position to intervene if we had any suspicions that anything was going wrong. Okay, so there's a rationale for this, and all three of these tests have a role to play here. So this is kind of the second take-home message. So if we have a patient with newly diagnosed disease, and we want to be able to predict how quickly the disease is going to grow and what other problems might arise, these are the tests that can help. So the FISH that we've talked about before, the immunoglobulin mutation status that we've talked about before, and obviously the p53 mutation status as well. And we can categorise patients into low or high risk of early disease progression or other things happening, and stratify their management accordingly. And these are obviously the features that, again, there's some jargon here that I apologise for, but there are things about the B-cell receptor that can help to predict the risk of things like Richter's transformation. You know what Richter's is? It's where the disease turns into a high-grade lymphoma and it becomes much more challenging to treat. And similarly, so that if patients don't have any of these things, we can make life easier for everybody by adapting the follow-up. Okay, so the third question is what to do once treatment has finished. Okay, and again, the same principles apply really. So if a patient has finished treatment, done well, if we can predict that patient is going to stay in a long remission, we can adapt the follow-up accordingly. We can see the patient less frequently or do the follow-up remotely. If the patient is predicted to relapse earlier, it's experimental at the moment, but these sorts of patients would be excellent candidates for trials of maintenance therapy. In other words, you've got the patient in a good place, but we know that things are going to hot up again sooner rather than later. Let's put some more treatment in to keep the disease in check. It's unproven at the moment, but this is those sort of patients are really good candidates for that sort of approach. And finally, if you've got a patient where you know that there's a high risk of a Richter transformation, even if the patient's in remission after initial treatment, that strengthens the case for doing something more radical with the treatment and considering, for example, allogeneic stem cell transplantation while you've got the opportunity to do so, instead of waiting for the disease to come back in the form of a high-grade lymphoma that might be really hard to control again. If you've got the patient in a good place so you can do a transplant, because we do transplants to keep patients in remission rather than get them there, then that can help with treatment decisions. Okay, so and this slide just summarises what I've just said really, but and of course some of the tests that can predict what happens after treatment you've already done before treatment starts, and you have to do before treatment starts actually, because there might not be any cells left after treatment that you can test. Okay, so the pre-treatment immunoglobulin gene status is useful, the pre-treatment FISH status is useful, and the pre-treatment p53 mutation status is useful. But there's an additional test that you can do after treatment, which is called minimal residual disease analysis. And what this means, it's basically a very sensitive way of detecting CLL cells, and you can do it in the blood or you can do it in the bone marrow, but it's more reliable if you do it in the bone marrow, especially if you've just finished treatment, because treatments tend to work better in the blood than the bone marrow. You can get an artificially good impression of the effect of the treatment if you just look at the blood. But basically, it's a bit like, it's exactly the same as I showed you at the beginning the technique of flow cytometry. where you stain cells with fluorescently labelled antibodies. Well, minimal residual disease assessment employs exactly the same technique, but it's a bit more sophisticated and much more sensitive, and it can detect 1 CLL cells in 10,000 normal white blood cells. And if you get down to below that level, you do brilliantly, basically. If you're above that level, you don't do quite as well, and there's a linear relationship. So the more disease you have left behind after treatment, the earlier it tends to come back. But you can actually measure it, you can actually quantify it using this technique. So the take-home message here: once you've done with the treatment, you've got all the test results that you did before the treatment was started. You know, the FISH, the immunoglobulin gene analysis, the p53 mutation data. You've got all that stored in the bank, as it were. But you can do an additional test after the treatment is completed, which is this MRD test, and then categorise patients into those where you can see a few CLL cells left over, and those where even at the 1 in 10,000 level you can't see anything. It doesn't mean they're not there. It just means that they're below that level of detection. And then you can adapt the treatment accordingly. So this slide, it's quite a busy slide, and I'm not going to go through it solemnly because it pretty much repeats everything I've just said, but it just summarizes everything I've said in one slide, basically. And yeah, so I'll just leave that there for you to digest. I'm nearly at the end now, so this is the last few slides, really. So there's a lot that we can do in terms of tests that can predict specific medical events that can help us advise you on treatment options, when to start, what treatment to give, what to do afterwards. But there's a lot that we don't understand still. There's been a real explosion in technology recently that has allowed us to look at the genetic code of CLL cells and other cancers and other diseases in much more detail. And what's come out of all this is the recognition that there are an awful lot of genes that are what we call recurrently mutated. In other words, they pop up time and time again. Not everyone has them. In fact, sometimes they only occur usually they only occur in a minority of patients, but they do. It's not just one patient. It's a group of patients. And I've listed some of them here. There are many more than that. Trouble is, we don't quite know what the significance of all these gene abnormalities is. What has become clear, and there's unpublished data from somebody, a PhD student in our group, is that despite all this, the dominant genes involved in determining outcome, at least, the really dominant one is still p53. So that's still the most important one, probably followed by this gene called ATM, and that's the gene that's missing on the long arm of chromosome 11, the 11q deletion. But so those are the two dominant ones, p53 especially. But you've got these other mutations. They probably all do something, but we're very unclear about exactly what they do and how important they are, and how they can help us make treatment decisions. There's a lot of ongoing work to be done there. So if we're thinking about in very practical terms about what we can do now in 2017, if you're a patient with CLL approaching treatment or having had treatment, especially early on during the course of the disease, these are a list of tests, I think, which a specialist CLL centre should probably be providing. And I've talked about all these, so I'm not going to go through them again, but immunoglobulin gene mutation status, FISH for chromosomal abnormalities, p53 mutation, and this minimal residual disease analysis. And I think in an ideal world, this is how we would wish to apply these tests. So at diagnosis, I think we need the first three to give first of all, to give you the big picture of how things are likely to turn out, especially if you've got early stage disease. And also to help us to advise on the timing of treatment, the choice of treatment, and what to do after treatment. So obviously, a diagnosis, lots of you won't need treating, but eventually you'll come to treatment. And then what tests should be done at that stage? So this doesn't need to be repeated because it stays the same. Okay, this is fixed. So you only need to do this once at diagnosis. You don't need to do it again. But these other things can change. So even if you've done them at diagnosis, there's a strong argument for doing them again before treatment because they can influence treatment decisions. After treatment, you can't do any of these because there's no cells there to do them with. But what you can do is this very sensitive test to see how much disease is left over. So you've got at diagnosis, before treatment, after treatment, and these 4 tests, and when I think it would be ideal to apply these tests. This is the final throes of my talk, you'll be pleased to know. So I mentioned before that we don't have all the answers. Our understanding of why the disease behaves differently in different patients is very incomplete. But there's a lot of work ongoing to improve this understanding. I'll just mention two things because both have a kind of Liverpool focus, really. So it was about in 2008, one of the working with Peter Hillman, who obviously chairs the National Trials Group, we established a biobank for CLL trials, which was based in Liverpool, and the idea was to have the opportunity to collect samples, CLL samples, from all patients entering clinical trials of CLL, so we could use those samples to understand more about the disease and its treatment. And it's been because everybody believed in it and supported it. It's been a tremendous success, and we've got a fantastic biobank now that's collected from all CLL trials going back. The ones coordinated through the UK group, at least going back to 2008 or thereabouts. And because of this, Genomics England. And the link there was with Anna Shu in Oxford. Anna's a great colleague and has been very collaborative, and is interested in genomics and study of the genetic code. Genomics England allocated £3.5 million to read the whole genetic code, not just the bits that code for genes, but the whole genetic code in up to 1,000 CLL samples that were stored in the biobank, linked to trials. And this was actually one of only 3 pilot studies for the definitive Genomics England 100,000 genomes project, which is now ongoing throughout the UK. So that was a real achievement for the UK CLL research community, and I'm very proud that that it was all based around the biobank that we set up in Liverpool. This project has been applied to samples from all of these trials, and I guess there'll be many of you in the room who have taken part in these studies. So that's very exciting, and we're just on the verge of getting some data out of all that. And the idea is that if for those patients who have contributed to this study, if there's anything. If anything is detected, and if there are any mutations that are detected that we think are useful in terms of your management, they will be transmitted to your doctors and with some accompanying advice. So that's really interesting and helpful. So the other thing I've just highlighted here is some work that again we're doing in Liverpool to go a step further than the genetic code. So in addition to having these samples that are characterised from the point of view of knowing every bit of genetic information about them, we're now looking at them at the next level, at the protein level. Okay, so we've got a technique that can simultaneously measure multiple proteins all at once. The proteins are obviously the building blocks that confer function. So they're kind of functionally more meaningful than the genetic code. And we're able to measure simultaneously the levels of about 7,000 different proteins all at once. And we're planning to apply this technique to the same samples that were analysed for their genetic code, and then link the two, so we can build up an understanding of how changes at the gene level affect changes at the protein level, and how that impacts on clinical outcome and response to therapy. So this is all very cutting edge, kind of exciting stuff. And there's all sorts of other research going on that I don't have kind of time to mention, and a lot of very active and successful research groups throughout the country, in places like Southampton, for example, and Birmingham, and Cardiff, and many other places besides. The point to make really is that this is the sort of stuff that is required to bring treatments on, but they require your support and participation in the form of clinical trials and sample collection. Without those things, none of this would be possible, and teamwork is therefore paramount. And this is my final slide, and it's just really an acknowledgement of all of the partners who have been involved in the UK's CLL research endeavours over the last 10 years, I guess. It's almost, well, I can tell you it's a very incomplete slide, and I apologise to anybody who's been part of this and hasn't appeared on the slide, but it gives you some flavour of the kind of teamwork that's involved in the different organisations that are involved required in order to deliver the sort of high quality practice changing research that we aspire to do. So on that note, I'll finish. Thank you.